Hello, this is Felix Drost from the Helmholtz Center Munich in Germany, and I will present to you MeTCRs, which is a deep metric learning approach for comparing T-cell receptors based on the sequences. Let's start with a motivation. Imagine a virus that will infect a body cell. This virus will be dissected into small peptides, so-called epitopes, which will be presented on the cell surface via the major histocompatibility complex. This complex can be recognized by T cells via the T-cell receptor, and the T cell can release upon recognition cytotoxins that will kill the infected cell. When studying T cells and T cell receptors for the development of biotherapeutics or vaccine, often the specificity is of great interest. We can infer this specificity by querying the receptor sequence against public databases of known T-cell receptor epitope pairs. Since identical sequences are quite constrictive, often we calculate pairwise distances between the databases and the query set of interest. For that, often string approaches are applied, like k-mer matching or added distances. However, the public databases now contain more than 150,000 T-cell receptors, and a single-cell study can contain more than 100,000 T-cell receptors as well. So pairwise distances via the computational heavy string approaches are often not feasible anymore. More recently, there have been variational autoencoder-based methods that have been employed to embed T-cell receptors and perform the comparison based on this embedding. However, they are trained unsupervised and therefore don't make use of this knowledge that is contained between these TCR epitope pairs from these databases. That's why we developed MeTCRs, which is a deep metric learning approach for comparing T-cell receptors. For that, we extracted the T-cell receptor information, especially the CDR3 region of the T-cell receptor, from these public databases, and embedded the sequence using one of these four architectures. While for most metric learning approaches, some sort of contrastive loss is employed, this requires negative samples, which are hard to obtain for T-cell receptors. This is why we employed the Barlow twin loss, which maximizes the cross-correlation between a pair of matching T-cell receptors, while avoiding the trivial solution via batch normalization. We benchmarked our approach and compared it against the baseline method, TCR match. Here we can see that for clustering and classification methods, the transformer-based method almost performs on par with TCR match, while still having all the benefits that come with a numeric embedding. When we take a look on the 5 most common epitopes in the evaluation dataset, we can see that here one version of MeTCRs at least almost outperforms the TCR match baseline method. This might be due to the heavy imbalance in the evaluation set, as well as the training set in regard to T-cell receptors, recognizing the most common epitopes. This brings us already for an outlook. As stated and as can be observed here in the epitope distributions and the UMAP, the evaluation and also the training set is heavily imbalanced. So we need to incorporate further ways during the training and the evaluation to tackle this imbalance. Further, we want to incorporate additional TCR information beside the CDR3 beta sequence, but this leads to a missing value problem. The method can be further improved by using large corpuses of TCRs with unknown specificity for unsupervised pre-training. On the evaluation side, we further want to expand our benchmark in a more database query setting, including a timing analysis, since this is the major use case. We further want to include more baseline methods like the commonly used TCR dist or deep TCR, which is an autoencoder based method. So to summarize, please stop by at our poster if you want to discuss T-cell receptors, especially for inferring specificity, or if you're interested in deep metric learning on biological sequences in general. In the end, I want to thank Leonard Schiefelbein for his great contribution to this project, as well as Benjamin Schubert for his supervision. Of course, I want to thank the funding agents, the Munich School of Data Science, the BMBF, and the Helmholtz Munich, without whose this work would not have been possible.